Hello. Hello in there. Hi. Okay, so we're doing a drawing on meiosis today. All right. So I'm gonna do a quick share my iPad and then we'll get ready hopefully for a drawing on meiosis. So let me oh, come on, share. Let's do this. Share. All right, so it might go into 2 pages, so beware. So before I start, I just want to remind you a couple things. We have mitosis and we have meiosis, right? So these are the two things that we're dealing with. Meiosis, chapter 12, mitosis is chapter 12, meiosis is chapter 13. Mitosis is cell division. Actually, let me rephrase. It is going to be nuclear division, nuclear division, and this creates 2 identical cells. We're going to see DNA replication followed by sister chromatid separation. All right. In meiosis, well, actually, I'm going to keep going. So for mitosis, this is for all somatic cells. Remember, somatic cells are all cells but your sex cells. And meiosis is to create gametes. All right, and the gametes are egg or sperm. All right, so here we're going to create this. Meiosis creates 4 different gametes. All right, so either egg if you're female or sperm if you're a male. So it creates 4 different gametes. We are going to see that the very first step in S phase is the same. We're going to see DNA replicate, but we're going to see two separations. The first separation, anaphase 1, is going to be homologous chromosomes separate. All right. So homologous chromosomes are going to separate first. And then we're going to see in anaphase 2, we're going to see sister chromatids separate. All right. And this is, again, only when we're creating gametes. Right. So they're fundamentally different. Mitosis, you replicate and you separate. Meiosis, we're going to replicate, separate, and then separate, right? And we get 4 gametes versus 2 identical cells. So let's go ahead and start drawing this out. All right, so we're going to have, this is going to be mom chromosomes are going to be here in pink for me because I don't know. It just helps me to remember who's who. And we're going to have dad. I know this is super gender generic. What's the word? Anyway, y'all know what I mean, but forgive me. I'm just going to go with it. So we have dad chromosomes and mom chromosomes. And here we are going to start with a diploid cell. And remember that a diploid cell is going to have 2 copies of every chromosome. All right, 2 copies of every chromosome. So for example, if we have 1 long pink chromosome, we better have 1 long blue chromosome because they're paired, right? So we've just drawn a homologous chromosome. And then if we draw a second homologous chromosome, then this is going to be small. And we have, for example, a small pair of chromosomes. So mitosis and meiosis start exactly the same. So here we are going to see in S phase of interphase that we're going to see DNA duplication. So every single chromosome is going to replicate, right? And they're going to be held together by their centromeres. And we're going to see a big one and a small one from both mom and dad. Okay, so that is duplicated chromosomes. So, and duplicated chromosomes create our sister chromatids, right? Okay, so the next thing we're going to do is that we are going to see, we're going to go through, I'm going to call it metaphase 1, right? Because we're going to have two separations in meiosis, they're separated into like 1 and 2. So this is the first metaphase. And if you recall in metaphase, something lines up at the metaphase plate. In mitosis, all the chromosomes line up so that you can split sister chromatids. In meiosis 1, we are going to see that that I'm going to put, actually, here, I'll put homologous chromosomes, or the homochromos, line up so that they will separate, all right? So what that means is that if we, or when we draw the metaphase plate, we better have homologous chromosomes that are splitting, all right? So first thing in this diagram here, let's label. These are the homologous chromosome. I'm going to call it the long homologous chromosomes. These guys are the short homologous chromosomes, right? So if we're going to line them up and we need to make sure that, for example, homologous chromosomes are going to separate, we need to line them up so that that is the case in our on our metaphase plate. So that means we would have the two big ones line up so that when we draw that metaphase plate, those would separate, and the little guys. Notice on metaphase 1, no chromosome is directly on the metaphase plate, right? Whoops, that's a big old microtubule. So, and again, they're going to be, the spindle is going to bind to the kinetochore on the chromosomes, all right? But is that the only way that you could line up chromosomes, right? And no, there's another way. So I'm just going to show you that there are multiple ways to line up homochromos at metaphase 1, all right? So I'll draw the other way. So if we have another one here, and again, I always like to draw my metaphase plate. That way I know where the centromeres are. And here we could, for example, have this top pair lined up exactly like that. However, we could also have a little pink one over here and the little blue one on this side. Those are both, so I'm going to label them here. So this 1 and 2, those are both perfectly good. Either way, we're lining up homologous chromosomes in line on the metaphase plate so that they will separate. They are identical, they're both perfectly good, right? And in fact, one or the other happens, but not both, right? And imagine then if you had 23 pairs of chromosomes, how many different combinations you might have in metaphase 1. This can lead to a lot of genetic diversity, all right? We're just going to keep going with this, with number 2 here, since it was our first one. And in anaphase, they will pull apart, and that will result in a chromosome here, a cell here that has only mom, and a cell here that has only dad. Most importantly, do they each have one big and one little, right? That's what's really important. So that would be after, let's see. That would be after anaphase 1, where they actually start pulling apart. And here we have telophase 1, where they actually split, right? For an example, if number 1 split, just to give you a sense, we would have, we would have different combination, right? So that leads to genetic diversity. So we're just going to continue with this one that we've been drawing. So let me redraw that on the next page. So what we have now is, so we just had telophase 1, right? And that resulted in 2 cells that are not identical, but they both have a duplicated big pair and a duplicated small. I shouldn't say pair. A duplicated small and a duplicated big, right? So what we did in the first metaphase is split homologous chromosomes, right? Now, we don't have any more DNA replication. We only have that at the very beginning. So then we are going to start lining them up at the metaphase plate again, all right? So let's say we have metaphase 2, right? So like if we're gonna line these up, what we're gonna see is that you want to line up so sister chromatids split. So sister chromatids. So let's say if I draw my metaphase plate here, metaphase plate here, then my chromosomes would line up so that our sisters would split. And over here, so that our sisters would split. All right. And then we would go through anaphase, where they are being pulled apart, right, by these centrosome spindle microtubules, right? They're going to be pulled apart in anaphase 2, rather. And then what is the result in telophase 2, right? So what we're going to see is that, let's just draw our chromosomes. So again, with this metaphase plate, we're going to see half going to this side. I'm going to draw them down here. So that means we have one big pink and we have one small pink, right? And on the other side, we're going to see the same thing. So we're going to see one big pink and one small pink. On the other cells, we have one big from dad and one small from dad, and one big from dad and one small from dad. So our result are 4 haploid cells. All right, and haploid literally means one copy of each chromosome, whereas diploid is 2 copies of every chromosome. Here we have 1 copy, one big and one small. And it was really at random whether we had like all moms in one and all dads are in another, because it really depended on this metaphase 1 event on how they lined up, right? So we result in 4 haploid cells, 1 copy of each chromosome, right? So main points, meiosis in metaphase 1, homologous chromosomes line up. And in anaphase 1, we see those homochromos separating, all right? In metaphase 2, we are going to see sisters, or actually, let me say, I'm going to put, we're going to see all chromosomes line up. And then in anaphase 2, we are going to see sister chromatids separate, right? So mitosis mimics more like anaphase 2, right? So that is the drawing. Hopefully that makes some sense, and I'll stop there. Peace. Bye.